So what we have seen over the last few decades is technology providing us with access to human architectural, structural information that allows us to build personalized brain network models. And for this, I like to give you an overview of how this has developed, in particular in, sorry, I'm trying to figure out how this works with a laser pointer, so I cannot point at this. Yeah. In the 90s, a major contribution was coming from magnetic resonance spectrography, magnetic resonance imaging, which has provided us with non-invasive access to structural and anatomical information. From this, a decade later, diffusion tensor-based imaging has allowed us to reconstruct the connectivity between brain areas in vivo. And as a consequence, this has allowed us to topologically characterize brain networks and actually span individual brain networks and co-register them with anatomical information, providing us with a skeleton, with a connectivity skeleton of individual brains. Hand in hand, this was going on with the advances in information communication technologies, high-performance computation theories, graph theoretical approaches to characterize these networks evolved and allowed us actually to compute them on supercomputers. This gave also birth to one of the European flagship projects, the European Brain Project, in which actually these approaches were taken and integrated in one common approach, leading to what we know today as EBRAINS, a digital neuroscience research infrastructure, which is the major output of the Human Brain Project. In these approaches, the data have started to drive the modeling efforts on the level of the full-scale brain network, which is now known as virtual brain modeling and has actually resulted in applications in clinical translation, in particular epilepsy. One clinical trial that is ongoing that I will speak about in a little more detail. So these technologies have driven the development of brain network models, and the vision is that we will be able to build what you find in the literature very often discussed today is the digital twin. So to build a virtual brain network that is informed by the individual's brain data. And this does exist today. These are the so-called virtual brains that are capable of generating patient-specific brain imaging data, e.g., magnetoencephalography, MEG, and functional magnetic resonance imaging. In this vision, we want to use these brain imaging models as in silico platforms to perform hypothesis testing, because you can actually use them to simulate brain imaging data and actually test clinical hypotheses. This can be used for either diagnostic purposes or for the development and application of therapeutic interventions. Yeah? And I'd like to illustrate this along the example of epilepsy. Epilepsy is one of the most prevalent brain diseases. 1% of the world population is affected. And 60% of epilepsy patients can be treated pharmaceutically. The remainder, 30 to 40%, there are other options, in particular brain surgery, the resection of the epileptogenic zone, that is the key area from which actually highly organized, synchronized oscillatory activity emerges and propagates through the network, handicapping the patient and the brain function. And you see here on the bottom right some of the signals that we are actually recording during pre-surgical exploration. This exploration is invasive. so-called stereotactic electroencephalography. So you're inserting EEG needles into the brain with 10, 15, or 20 electrodes, in which you can actually record the ongoing activity in the brain. Typically, the patient keeps this during 2 weeks in order to be able to be followed, and so we can actually extract the information about the organization of the seizure activity. This technique has been developed in the 60s, 70s in Paris by Jean Bancourt and Jean Talarac, and this has become one of the standard techniques for pre-surgical exploration. Notable in this context is that surgery success rate has not improved over the past 40, 50 years. And one of the key reasons for that is that we are still not able to correctly identify and outline the epileptogenic zone, which is actually the target for the resective surgery. What we do with the virtual brain approaches is that we build a virtual brain template for a particular patient, then use it as a target for fitting of individual parameters that become personalized, and these parameters are representative for epileptogenicity in the brain, in the case of this particular application. And there we are taking full use of novel methods in artificial intelligence, in deep learning, and in particular Bayesian inference. The way the workflow is organized, you see here on the right, you have a fusion, an integration of different data streams. On the one hand side, we use the structural information of cortical and subcortical structures, and we parcellate it, which provide us with the network nodes, which you see in color-coded here, at the, where it's written VP, Virtual Epileptic Patient Atlas. On the other hand, we co-register it with diffusion-based imaging, where we reconstruct the connectome, co-registered with the nodes. The connectome provides us with the links, and that allows us to span the network in three-dimensional physical space. This is what you see in the middle. Yeah? And then from the computer tomography, CT, we get the positions of the electrode needles, of the SEEG electrodes, and again co-register this in the same brain space. And the individual electrodes, these are the red dots you see here, are actually the places where the activity is registered. So we're talking about sources on the one hand side, these are the yellow dots, and the electrode points where the sensor signals are being registered. We equip the sources with mathematical models, many of those that have been developed over the last 7, 8 years in the Human Brain Project, where statistical reductions are being made of populations of neurons. We understand the activity of neurons quite well, but now we use mean field theory to scale it up to reduced representations, so-called neural mass or neural population models, that are reduced representations of brain region activity. We equip these yellow balls with these brain regions with these models and can now build a network and actually perform the simulations. Using the methods of artificial intelligence and deep learning, we can actually now estimate the parameters of these models, which are the parameterization of an individual brain model. And the target is to identify the epileptogenic zone. So once the model is parameterized, we have an in silico platform. And you see a simulation here of a patient's own brain model. In blue, the sources. In yellow, this time, the electrodes. Down on the bottom, you see how the seizure evolves. And this is purely simulation. Once the epileptogenic zone has been identified, we can run on the high-performance computing structures these models and simulate the seizure propagations and their variability, because intra-subject variability is one of the big challenges that we encounter. But please note that we have the information on the source level, which is very important, and on the SEEG level. What I'm plotting down there are actually the information on the SEEG. So on the sensor level. Yeah? So this type of technology, on the one hand side, is a target for fitting, so diagnostic purposes. On the other hand, as an in silico platform for simulation, is being used prospectively in a large-scale clinical trial that is ongoing, started in June 2019. You see here the patient inclusion numbers. You see also the first lockdown in France that we had in March 2020. Slowed down, but we were able to continue. The target is in 4 years to obtain 400 patients across 13 reference centers in France, of which half of them will be virtualized. And this extra information is being provided to the clinical staff for additional decision-making. Yeah? So there are 2 more years to go, but retrospective data already exist. There are different types of evaluations that can be performed. Here in this example, for instance, you see a patient before surgery and after surgery, and in the red plotted areas is before surgery, the identified epileptogenic zone as target of the resection, so overlaid with a post-surgical MRI that is represented here. And this you can compare now, these results, you can compare now against a clinical standard of care. Typically defined 12 months after surgery, seizure freedom or not. This can be quantified. There is the Engel classification score. And then you can actually compare it against other metrics, against a clinical hypothesis. And what you see here on the right is actually precision, which is a statistical score, similar to specificity, yeah? That actually, on the right-hand side, you see VP, yeah? How it drops down when it's actually, and this drop in VP performance is significantly related to negative surgery outcome, indicative that there is a correlation with positive surgery outcome if the surgery had been followed. Yeah? But there are many other evaluations, but this is at the moment entirely correlational because retrospective. The prospective clinical trial will tell us more. The question is, what actually matters in these approaches, in this type of modeling? Yeah? Is it the data-driven information that is coming from the imaging? Or is it the mathematics? Is it the deep learning methods that we are applying there? And the answer is actually multiple elements. And this you can systematically test in animal experiments. Here in a study with 19 mice that have been scanned, you can actually reconstruct the mouse brain, either using highly detailed data, for instance, from the Allen Institute, the mouse atlas with all the connectomics, or you can scan the mouse as we do it in the human and reconstruct the mouse's own connectome. Systematically, when you do this, the consequences and the results are unambiguous. High resolution matters. Yeah? So ex vivo information as obtained from the Allen Institute provided the best predictive power, almost approaching scan-rescan variability of the brain activations that were measured, which is in this violet color that you see there. Yeah? However, individuality of the connectome, its own connectome, also matters. So these are aspects we need to get together, but they are orthogonal. One is ex vivo, the other one is in vivo. So we need to build actually approaches that allow to fuse this. Yeah? And this is where it's going next, beyond the classic virtual brain approaches that exist today. One approach is high resolution. We need to get access to high resolution data, either ex vivo or in vivo, but in vivo it will be typically impossible, in order to build detailed meshes of cortical and subcortical structures and link it to connectivity. And here you see on the right-hand side representation of what is ongoing, scaled up by a factor of 1,000 to what is being done in the clinical trial, where we have 100, 200 nodes. In the high-resolution representation, we talk about 260,000 nodes, getting us to the millimeter scale. And this is a scale that you actually need in order to have a faithful representation of electric fields, as represented on the right-hand side. This increases the error and improves the identifiability of the estimation, but also it opens up the park and the avenue for novel application of techniques such as brain stimulation. And this is because you can actually not only implant a deep brain stimulation electrode and then map faithfully the electric field, or as represented here. You can use also novel techniques that are non-invasive, such as transcranial temporal interference stimulation that is focal, but needs a good representation on the cortical subcortical structures. This is becoming possible now, technologically at least. Yeah? So, data fusion will be needed, and for this, approaches need to be developed to actually get this ex vivo and in vivo information together. Hierarchical Bayesian inference is one of the techniques that is being used for that. In order to get this to the patient, there are steps in between. I spoke about invention, translated to innovation, but there's still a far way to go further to get it all the way to the patient. And for this, efforts of all of us are being needed, and here I would like to point out one that is linked by some of the institutions that I'm involved in, who have actually created a spin-off called Virtual Brain Technologies just a few months ago, the CEO Jean-Marc Ferrier being here in the room with us. And the idea is actually to take this technology, support and emphasize the development, and then get it all the way to the patient. On the right-hand side, the roadmap is being laid out. Take the existing VEP technology and scale it up to high resolutions. This will become the first minimally viable product with an aspired market access in 2025, 2026. Yeah? When linked to brain stimulation technology in the high-resolution environment, as I just showed you, then it opens up the possibilities to go to many other diseases and applications, such as diagnosis or therapeutics. Yeah? So the objectives are fairly standard, but very importantly, such as regulatory and market access strategy developments and finding the appropriate finances for this very capital-intensive projects. But beyond that, what is needed are educational approaches to to learn to use this type of tools and make it part of our culture in the application, in the standard of care, and the way how it shall develop to. Yeah? I stop at this point here. I would like to thank you. I would like to thank my colleagues and friends and the clinical team, in particular Fabrice Bartholomé, that is represented here, and the institutions that have supported us. Thank you.
Thank you. Thank you very much, Victor. All right, thank you, Victor. And I guess you'll receive plenty of questions during the break to you and Jean-Marc. So we invite you to interact with Victor if you have any question on the presentation during the break in the exhibition village. But Victor, I would have one for you because you mentioned the different approaches in the virtualization of brain models. But what is the key distinguishing feature of the virtual brain technology that you just presented?
Well, in the diagnostic domain, when you compare it to other applications, so to be very concrete, in epilepsy, the estimation of the epileptogenic zone, all the other approaches provide you with just another metric that is estimating the delineation, the organization of the epileptogenic zone. Given the generative modeling approach of the virtual brain, you can actually start interrogating this metric that is being provided by the virtual brain. In other words, you can also provide an estimate of the epileptogenic zone, but now you can start questioning it, because in the simulations, you can, for instance, implement this clinical hypothesis on the epileptogenic zone, run simulations, see the variability of seizures you get, and then you can change it, enhance it, make it smaller, change the topology, perform a lesion in there. So you can systematically interrogate the result that you have, and this adds a completely different value to this metric because you can challenge it intellectually and clinically.